Hello everybody! Today we are going to focus on the results from a CGH array with non-pathogenic SV and regulatory elements overlapped. So let's focus on this case study with a prenatal diagnosis using a CGH array. This study was performed on a fetus with retroagnathia on cleft palate. These symptoms can be standardized with two HBO terms, as you can see here. And this CGH array revealed 10 SV, 4 duplication and 6 deletion. So we submitted to AnnotSV these 10 SV coordinates and SV type in a bed format with 4 columns, chromosome, start, end, and SV type. Here is the output in compact mode with the 10 SV lines. As you can see, with the HCMG class, just here, the top line is the only one classified as pathogenic, and the other SV are all classified as VOS, variant of unknown significance. So, okay, let's focus on this deletion. There are different key questions to answer. First question, how many genes are overlapped? Well, we can see that 23 genes are overlapped. Are there morbid genes? Yes, when you move the mouse over the OMIM annotation, you can see that there are some morbid genes. Are they close to the phenotype? Well, the exomizer score is quite high because it's upper than 0.5, but it is lower than the threshold of 0.7 to be really specific to the patient's phenotype. Okay, next question. Is the deletion overlapping already non-pathogenic or benign SV? Actually, yes. As you can see, some pathogenic SV are overlapped with the deletion, but non-benign SV overlapped the deletion. Next question. Are there any regulatory elements overlapped? What about their gene target? Are there morbid genes or genes close to the phenotype? This point is very interesting because the regulatory element of the SOX9 gene here is overlapped, and this is a morbid gene that is called 0.8 with the exomizer module, just here. So these can explain the patient's phenotype. Finally, last question, what is a computed ACMG clean gene class and why? This deletion is classified as pathogenic. And if you move mouse over this field, you view tooltips with the AnnotSV ranking criteria. So here in the tooltip, you can see all the criteria of this classification, which correspond to those we have just spoken of. So if we click on the AnnotSV ID link, we can visualize the deletion highlighted in blue in the UCSC genome browser, and we see that the deletion is not overlapping the SOX9 gene, indicated here with a red arrow, and we also see that the deletion encompasses non-pathogenic clean gene on OMIM SV close to SOX9. So to conclude this case study, AnnotSV revealed the existence of a deletion close to the SOX9 gene. The regulatory element of the SOX9 gene is overlapped, and thanks to the phenotype-driven analysis, we know that the SOX9 gene defect explains the patient phenotype. This case study illustrates how the annotation of regulatory elements can identify causative variants as well as pathogenic SV overlapping the SV of interest. So, hope you liked this video. Thank you for watching, and bye-bye.